हाँ, 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 हाँ। ठीक है, ठीक है, ठीक है, ठीक है। ओके। Can you see my screen now?
No, ma'am, it's not visible.
हेलो? मेरी स्क्रीन दिख रही है? की है मैंने अभी। I think now you can see. Okay. हम्म, फुल स्क्रीन है। हो गया, हो गया, हो गया। हम्म। ओके। I'm not able to hear from the other side, but hopefully you guys can hear me. Can anybody respond? Because I'm not able to hear from any of you. Shall I start? Shall I start? Okay. So first of all, I would like to thank the organizer for inviting me for this talk. And today I'll be talking about the various trends and advances that have been taken place during the course of time in the area of diagnostic technologies. So, my talk would be basically focused on the RNA viruses. So, I'll be talking about some of the advances that have taken place in the area of diagnostics for uh, infections of due to RNA viruses. So, if we talk about the RNA viruses, they cause almost one third of the identified species and the diseases that have caused the human threat. So, in the healthcare industry, the global threat have been there since long, and the examples include the influenza virus, HIV, SARS-CoV, Ebola viruses, Zika viruses, polio viruses, and all. So we have been using various kinds of remedies to eliminate or for the remedy of these viruses, but some of the viral infections have taken us to the pandemics, and the recent one was the COVID 19. So, if we talk about the RNA viruses, they are not the recent ones. They have been causing different kind of pandemics since long, and we know that these RNA viruses, they have caused several threats to us, several diseases, and which include the common cold, the bird flu, and COVID 19, Ebola, and all. So, this is not new. But the problem is we have not been able to detect these viruses for the causative agent for these kind of infections. So, in the traditional use, we have been using the clinical samples for clinical examination, microscopic serology studies, ELISA techniques, immunofluorescence, Western blotting, and all. So, we have been using these traditional approaches to diagnose the causative agent like RNA viruses. And what are the challenges behind this? If we talk about the beginning of the century, since 1930s to the recent pandemic of COVID-19, we have been using different approaches, which include the gene chips, loop mediated isothermal detection amplification techniques, PCR techniques. So these are the molecular techniques. However, serological techniques have been uses since last several years, and there have been advances in the serological techniques, which include the immunoglobulin and ELISA techniques and enzyme immunoassay ELISA techniques and all. So including this, direct cell culture have been used in the since beginning. So there are the limitations for each one of the techniques that we use, and with due course of time we have been able to come up with some of the techniques which have helped us to save time to detect the causative agents. And if we talk about the biosensors have been able to help us in the diagnosis of RNA, the diseases which have been caused by RNA viruses. So, various electrochemical, optical, piezoelectric, and microfluid biosensors have been used, while they help us with the high sensitivity and the less time consuming, so real time data can be achieved, and various rapid techniques and cost effective techniques have been available with the help of the biosensors. And if we talk about the recent advances, there have been some innovations in the area of biosensors, which include the CRISPR-Cas systems, nucleic acid hybridization system, isothermal amplifications, and RNA aptamers, which have helped us to develop new kind of biosensors, and they have helped us not only to save time, but they have also helped us in new approaches which have helped the diagnostic industry a lot. So technology wise, as well as cost wise, as well as time wise, they have helped us. So if we talk about how these biosensors can have innovations, so there are chances to enhance the stability of these biosensors. We can have the wearable biosensors, non-invasive systems, which can help us for the detection of these viruses. And if we talk about the point of care technologies, they have been examples include electrochemical biosensors, lab on chip, surface plasmon resonance, and several others. And they have been proved to be very rapid, cost effective for on site testing. But they have also been useful for the um, where the resources are limited, so they can be taken to the site, and when the patient is not able to move to the diagnostic center, so these can help very much for the sensitive detection of the causative agent. For example, in the pandemic there were lot of limitations for the movement of various people, and it would have been very easier if we could help these patients with the point of care devices. So RT lamp is one of the recent one which has helped us to detect the viruses, where if the sample is added to the device, it can directly detect the qualitative test. So positive and negative can be done very easily with less time consumption. And the historically serological tests, cell culture and electron microscopy have been used, and the latest techniques have been with the use of molecular techniques, so that include the RT-PCR, then amplification through microfluidics. So lamp is, as I said, is one of the latest one. So they gave the faster results, they gave improved access as well as the enhanced patient care. And the point of care assays, if we talk about, they have the, if we can identify the biomarkers, they can be proved to be very effective. So for viral proteins, it can identify the specific proteins which are expressed by RNA viruses. There are targets which where we see we can identify the RNA, particular RNA sequences, and host human response can detect the antibodies. So these are some of the uh, approaches which have been used for point of care. So RT-PCR, as we know that this amplifies the specific the sequence, and if we have the gold standards and we have the method, then we can detect, but it is very very time consuming. So CRISPR-Cas technique has been taken as the advantages because it employs the target approach. So Sherlock and technique is one of the highly sensitive one, and if we can integrate with the portable platform, it can be very very effective. And then lateral flow assay. So lateral flow assay, why it is very important? Because if we add the sample to one side, the fluid can flow in the lateral manner, and it can prove to be very rapid, cost effective, and it can provide the visual results, so that the person it can be used by anyone which are not very very technically sound, and it can be used for the qualitative detection. So there have been some advances where smartphones have been used, and if we use the lateral flow assay, and the result can come with the smartphone, so it can integrate the microfluidics and biosensors. If we can integrate, the results can come in the smartphone also. And if there are cameras and all, so these are some of the integrated approach which people have been trying in due course of time. So microfluidic approach on and the lab on chip approach, this is one of the recent ones, and it uses the miniaturized process where the sample preparation, amplification and detection can be done in the on the same device. And it is, since it is compact, it will not cost very much, and it can detect rapidly, and the decentralized settings can be used for these kind of devices. So as you can see, if we add the sample, the DNA, the nucleic acid can be extracted, and with the lateral flow, amplification and detection can be done to give the output. And the if we talk about the modern diagnostics, so it can have the rapid response with the high sensitivity and specificity, with the cost, and as well as the resources. So the device, small devices can be taken to the anywhere, and can be very effective point of care devices. So there are microfluidic devices, CRISPR techniques, AI driven techniques, and they have been tried by various researchers, and the integrated approaches have been suggested by the researchers. And if we see the recent patents that have been filed for these devices, they have proved to be the effective ones for point of care devices. So that's all for the today's presentation, and this is the technique. Somehow there is a slide missing. So recently we have developed a device for the detection of RNA viruses, where we have seen that the microfluidic lateral flow assay, lamp assay, has been used for the RNA virus detection, and we have tested it with the various samples that were collected from the hospitals, and it has proved to be very very effective. And we have seen that more than 80 to 90% one is very effective. So that's all from my side, and hopefully I could do justice. I am not able to hear any one of you. Maybe someone is calling me, and I'll just pick up that call. And hello. uh, Hello.
हाँ ठीक है। हेलो। नहीं नहीं, म्यूट नहीं है। ऑडिबल हूँ कि नहीं? Yeah, thank you very much. Am I audible? Am I audible? Okay, thank you very much for a nice introduction. So I'm very much thankful to, yeah, sorry. I'm very much thankful to organizing team of BBIF 2025 for giving me this opportunity to present virtually my work. So today I will be talking about molecular imprinting based sensing strategy for detection and analysis of vitamin B12 by assessing holo TC levels in blood. So first of all, I would like to introduce about. Uh, I think slides is not moving actually. Slide is not moving. Yeah. Okay. Yeah. Am I audible? Am I audible? Okay. So this is just a brief introduction about vitamin B12. So this is structural detail of vitamin B12. Here we can see this is a porphyrin biomolecule having corin ring uh, containing biomolecule which has cobalt atom in the center, and it has six bonding sites, so four of which are actually bound to nitrogen of the corin ring, and the fifth is bound to fifth 5,6 dimethyl benzidamide, and sixth is which basically in this figure it is represented as R, and this has choice of many molecules. So in case if it is cyanide, CN, so this would be cyanocobalamin. If this group is CH3, then this would be methylcobalamin. So this is a complex structure of vitamin B12, though it is a very important vitamin. The main function of this vitamin is it is needed for myelination of nerves, act as coenzyme for various metabolic reactions, and it is required for formation of red blood cells, DNA replications, and its deficiency is very serious as it leads to pernicious anemia, bone marrow failure, degeneration of spinal cord, and hyperhomocystinemia. So because it is complicated vitamin, and its deficiency is also very complicated, so that's why its detection is only it is very important. So one of the major cause of vitamin B12 deficiency is dietary insufficiency, because the major source for this vitamin B12 is animal source. So mainly the vegetarian population is suffering with the deficiency of vitamin B12. And if we look upon the data, 15 to 20% of elderly population in United States is actually suffering with insufficiency of vitamin B12. 36% in Germany are present with insufficient levels of vitamin B12. If we talk about the Indian scenario, so 47% to 71% adult prevalence is actually highly deficient in cobalamin. So now, a brief about the metabolism of vitamin B12. So, when we take vitamin B12 in diet, it is bound to the protein, and after taking it in the stomach, it is actually digested with the help of gastric juice, and the protein part is digested, and cobalamin is released. And this cobalamin then binds to heptocorin, which is again a protein which is secreted from the parietal cells of the stomach, and this heptocorin bound cobalamin then reaches to duodenum, where again this is digested by the proteases, and it then bound to intensive factor, which is again released from the uh, stomach. So this intensive factor bound cobalamin is very important form, because this is the form which would be actually absorbed by the breast border lining of the intestine. Otherwise cobalamin cannot be absorbed by the body. So after binding with intensive factor, this is then released into the blood. So intensive factor is goes back to the system uh, in basically in duodenum. So then after that, this absorption of the cobalamin, 80% of this cobalamin again get bound to heptocorin, and only 20% total cobalamin, 20% of total cobalamin is bound to holo TC. It is actually a form which is actually available. Holo TC is nothing but a protein which is actually available for the absorption of, which is actually available for the various functions of the cells, like metabolic functions and other functions like cell repair or uh, DNA repair, etc. So there are the four main markers for for the estimation and detection of vitamin B12 levels. First is total serum vitamin B12 levels. Second is holotranscobalamin, homocysteine levels, and methylmalonic acid level. Homocysteine and methylmalonic acids are a good biomarker for detection of vitamin B12 deficiency, but homocysteine level basically are associated with the folic acid deficiency also. So this might create a confusion that the elevated level of homocysteine could be maybe because of vitamin 2 B12 level or because of the folic acid deficiency. And other methylmalonic acid level are also elevated in some cases, like in renal failure also, because methylmalonic acid is produced in other metabolic activities also. So here we are targeting about holo TC, or we can say holotranscobalamin detection, which is a novel biomarker. It is proven in the literature that this, because this is the form which is easily available for the cell or body for the metabolic functions. So we are targeting this uh, vitamin B12 detection using this particular biomarker. And total serum vitamin B12 level actually is the test which is currently run in the pathological labs. So next, the estimation of vitamin B12 level. These are the tests which I have already mentioned: methylmalonic acid test, radioimmunoassay, total vitamin B12, HPLC, ELISA. So these tests are sensitive, but they are coupled with some drawbacks like time consuming, required specialized skills for operation, tedious, sample preparation, expensive. So these all drawbacks can be sorted with the application of biosensing technology, as these are uh, known as ease for the operation, and cost effective, efficient, highly sensitive, and rapid also. So you all know about the biosensor because Dr. Asa Chaube has already introduced about the biosensor, so I will not spend much time on this slide. Though these are the various parts: biorecognition elements, immobilization interface, and transducer uh, of a typical biosensing device. So in this case, biosensing element could be antibody, aptamer, cells, enzymes. But in this study, we have not chosen any biorecognition element. We have chosen basically a phenol, or we can say molecular imprinted polymer. So that polymer will act as a biorecognition element in detection of holo TC. An immobilization surface could be anything. Anything means like biocompatible nanoparticles, cross-linking agents, or some biomolecules can also act as a bioimmobilization interface. And transducer is the part of the biosensor which actually convert biological phenomena into measurable electronic signal. So here, majority in majority, three types of transducers are actually used by population: electrochemical, piezoelectric, and optical. So we are focusing on electrochemical because these are easy to fabricate, highly sensitive and robust, and they have potential to miniaturize. So here aim is molecular imprinting based detection and estimation of whole OTC using antibodies which are coupled with thionine. Thionine basically is a protein which is found in higher plant species, and this has electroactive property also. So because of that, we have coupled this antibody, and because uh, this makes this strategy novel, so we have utilized this molecule. uh, so if we talk about the synthesis and fabrication, so first of all we have synthesized GIF8 particle. GIF8 is nothing but this is basically a organic metal nanoparticle framework. So here zeolitic imidazole framework was basically uh, made using hydrothermal method, and fabrication on supporting materials such as FT or ITO can be done. And then electro deposition of gold nanoparticle was done on GIF8 modified FTO, and followed by immobilization of template molecule. And before that, cysteine was immobilized to successful immobilization of holo TC onto this particular electrode. And then imprinting site of holo TC were created using phenol as an imprinting polymer by with the help of cyclic voltammetry. And this whole system was actually assessed or detected with the help of thionine conjugated antibodies to confirm the presence of holo TC after site formation. So here, this is the working strategy how we have made this MIP based sensor for holo TC detection. So in the first step, here we can see this FTO electrode is there, and onto this GFET uh, framework was immobilized, and followed by the attachment of gold nanoparticle. And in third step, you can see uh, there is immobilization of cysteine, which is amino acid, sulfur containing amino acid. And after attachment of cysteine to this gold nanoparticle, because gold nanoparticles have affinity for sulfur, and sulfur is present in cysteine, so that's why we have utilized this cysteine for as a linker. And EDC-NHS activation was done to immobilize this holo TC protein. And after that, polyphenol phenol was actually electrodeposited in fourth step by uh, cyclic voltammetry. And after for 30 cycle, I think for 30 cycle the electrodeposition of this uh, mm, polyphenol was done. And after coating of this polyphenol, the extraction of this holo TC protein was done with the help of acetic acid for around 40 minutes. And after that, again the incubation of this holo TC protein on this holo TC removed MIP sensor was done to check the cavity is made or not, or if the cavity is there, that the cavity can fit the holo TC molecule properly or not. So by assuming that holo TC molecule is there, then after that we used this thionine conjugated antibody. And because this antibody is highly specific to holo TC, so it would bind to holo TC only. If holo TC is there, then this antibody would be there, or if this antibody is there, then only we will get the signal of thionine. So this is the working strategy for the estimation of holo TC in serum. So next, this slide includes the characterization studies. Here we have done the SAM images and EDS spectra to check the presence of various elements. So in SAM pictures, we can see first is of GIF8, and after the GIF8 structure is then modified after the deposition of gold nanoparticle, and this is further modified when cysteine is attached to this. So, and after holo TC incubation onto this cysteine modified gold nanoparticle GIF electrode, there is presence of some globular structure in this fourth uh, picture. So we are assuming this is because of the presence of holo TC. And in the fifth picture, MIP is done, electropolymerization of phenol molecule. So after that, the sixth picture is basically belonging to uh, conjugation of this antibody to holo TC attached cysteine gold nanoparticle GIF8 electrode. So this surface modification actually ensures that we are going in right direction during the fabrication of MIP sensor. And this B's picture is basically related to EDS spectra, and this presents the uh, various elements which are present in uh, gold nanoparticle GIF8 FTO surface. So presence of gold, zinc, oxygen, carbon, nitrogen, these all confirms the successful formation of this gold nanoparticle dot coated GIF8 FTO. And this is related to characterization studies. Here we can see first picture is of GIF8, then followed by gold nanoparticle attached GIF8, and then cysteine. So in third one we can see that there is a in in figure A, yellow graph shows 3286 per centimeter peak, which is because of basically the NH stretching. NH stretching is because of the presence of amino acid cysteine. So this cysteine is actually having NH2 group. So because of that, we got this. And other peaks are also because of the presence of cysteine. And after attachment of holo TC, which is again protein made up of amino acids, so this peak then get dense because of NH stretching. And other peak are also there, which are actually related to amide 1 and amide 2 peak bond of the holo TC. After that, MIP was formed. MIP is basically the polyphenol deposition onto the surface. So, because of the MIP formation, the peak holo TC was masked and cysteine was masked. So, that's why these all peaks are actually uh, lowered. So, they are less presence of these peaks. And after the attachment of uh, antibodies which is conjugated to thionine, there is again presence of 3310 centimeter peak, which is because of I'm so sorry, just a second. So and because of this, there is again presence of 3310 centimeter peak because antibodies are again made up of protein, and because of the presence of protein, NH stretching is again in picture. The C graph is basically related to contact angle studies, where we can see that during the each step of modification of MIP sensor, we can see the change in the hydrophilicity of the surface. First is of GIF8, and after deposition of gold nanoparticle, the hydrophilicity increased, and further hydrophilicity increased when cysteine was attached. And when holo TC was attached to this surface, there was increase in contact angle up to the 68.5 degrees Celsius. In the fifth picture, we can see then when MIP was made, the contact angle further reduced to 42.8 degree. And there was, after the holo TC removal, the contact angle or wettability of the surface was again increased up to 38.8 degrees Celsius. And after attachment of antibody, which was conjugated to thionine, the uh, the hydrophobicity of the surface increased up to 80.9 degree. So this actually confirms the successful formation of MIP sensor. This is electrochemical characterization studies, where we can see the first graph in the A picture. The yellow one is of the DPB analysis of GIF8, and after the attachment of gold nanoparticle, the peak spiked at zero point 0.2 nanometer, and then after attachment of cysteine, there is further increase in anodic peak current, and after that the holo TC mobilization, because holo TC is a protein, a bulky structure, and a non-insulating, insulating, also not conducting in nature. So that's why the peak current decreased here in green graph. So and after MIP sensor means the there is a polymerization of electropolymerization of phenol onto the surface of this holo TC attached electrode. So there is a again there is a decrease in the current here in case of this B picture, B, and that after removal of holo TC, the current further increased. So because holo TC is not there, so that's why the surface is again conducting comparatively. And then because of the presence of cysteine, because the site are created, and so that's why there is the presence of point 0.2, a peak at point 0.2 volt. And because of this confirms that there is successful removal of holo TC. And when holo TC was again rebinded, then there was further decrease in the peak. So it is actually related, the MIP sensors peak and after holo TC rebinding are the same. And this is NIP sensor, which is basically non-imprinted sensor. Here holo TC is not present, rest steps are same. So these are DPV graphs, and these blow graphs C and D are actually CV graph of same electrodes, and these results are in sync with just a second. Yes, am I not audible? Oh, okay, just give me 2 minutes. 2 minutes. Okay. So after this, the electrodes, these various optimization studies were done, and then electrochemical sensing of holo TC was done using DPV. So linear range for this was zero point zero 0.01 picogram to 100 nanogram, and limit of detection was this. So we got very nice detection limit, and this is the first report. It is not there is no report on the detection of holo TC using MIP based biosensor, and this is related to selectivity, stability, and reproducibility. And this is highly selective sensor, and the stability is also 40 days at 4 degrees Celsius. It is because we have not used any biomolecules, so that's why it is there. Otherwise because biomolecule have tendency to get denature at normal temperatures. And this is real sample analysis. Here we took the four blood samples of healthy individuals, and these were tested with routine total vitamin B12 test, and by using our MIP sensor. So the detection, the limit of the range in which holo TC was detected was 4.6 to 15.6, because 6 to 20% fractions of vitamin B12 is bound to holo TC, and our sensor was able to detect this concentration 14.1, which is actually lying in the range of this. So it means our biosensor, the developed biosensor, is actually working fine, and further attestation is still needed. So these are the conclusions. So quite good detection limit, and this is the work which we have already published in biosensor and bioelectronics. And in comparison to this previous work, this work is actually have better LOD, and shelf life is also good, though response time is little high. So thank you very much for giving me this opportunity to present my work. And acknowledgement is basically to my current research team. Rishika Rohilla is the main PhD scholar who did this work, and Amanpreet Kaur, Sonia Rani, Surbhi, Sarita Galyan, and Punjab University, who gave me this platform to carry out this research work. Thank you very much. Thank you. So any query? I apologize as I went beyond the time. Am I audible? हाँ जी बोलिए। अच्छा। ओके, थैंक यू। सो ओके, ओके, थैंक यू। आई हैव ऑलरेडी डन माय लंच। थैंक यू सो मच। थैंक यू सो मच। थैंक यू। बाय।